I am Dr. Sathya, working as an assistant professor in Department of Chemistry, SNS College of Engineering. Today we are going to discuss one of the absorption spectroscopy. It's called IR spectroscopy. So what is meant by spectroscopy? When electromagnetic radiation is passed through the sample to emit some radiations like EMR radiation, UV visible, IR, microwaves, gamma rays, so a lot of the rays. In general, the UV visible 200 to 400 nanometer, for visible 400 to 800 nanometer. So after 800, the infrared region is called wave number. So the measurement of the interaction of infrared radiation with matter by different types. One is absorption, emission and reflection. This is IR instrumentation diagram. Here the IR source is nurse glare or L gobar. The two types of the samples source used in IR instrumentation. This light source will be passed through the monochromator. The monochromator will be give some polychromatic radiation to give the narrow band of radiation. It's passed through the mirror. After that, the mirror through the light will be interact to the sample cell and reference cell. And finally, the splitter will be split the light radiation to give the detector, and finally we got the result in the recorder. So this is the IR instrumentation, the basic things. In IR source, the monochromator, sample cells and reference cell. Here the sample cell and reference cell, this reference cell is used for here which solute compounds is soluble in solvent. So that solvent is called at the reference. So near IR spectroscopy, it's a lot of bio applications. Near IR spectroscopy also one type of the IR spectroscopy. This is the electronic transition of IR spectroscopy. Here the rotational and vibrational energy levels, it will be go into the higher energy level. In IR spectroscopy, the molecules is always the vibrating things. Vibration means different types of vibrations. The molecules are different stretching and bending vibrations. In rotational energy level, the molecules will be ground state to excited into the excited energy will be transition to the excited state. So this is called electronic transition of vibrational spectroscopy. So the region of IR, so the different region of IR is present in that IR spectroscopy. Near IR, the wavelength will be listed here. Here the wave number, the different types of frequencies or interact with the near and middle, far and IR. So this is symmetric stitching. Different types of stitching. One is the symmetrical, anti-symmetric. So symmetric stitching, the oxygen will be stitched into opposite side. Okay, the oxygen will be stitched into through the carbon towards the carbon atom. So this is the symmetric stitching. In asymmetric stitching, the carbon with the carbon atom, one oxygen towards the carbon atom and other oxygen away from the carbon atom. This is called the anti-symmetric stitching. The next two animation diagram is the bending. So two types of bending available for IR spectroscopy. So the symmetric stretching and asymmetric stretching and rocking, wagging, twisting, scissoring. So these are the types of IR spectroscopic vibrations. The scissoring is we are using scissor, no, that like will be cutting into the like this. So the two mirror atoms will be vibration like this. So this is called scissoring. Wagging. Wagging means just movement of the two atoms. This is a wagging. Twisting means the two atoms is twisted to one another. This is called twisting atoms. This is also one of the example for symmetric stitching and anti-symmetric stitching. So already we discussed in previous slides, and also we know about that oxygen, the two hydrogen atom will be stretching away to the oxygen atom. Here the one hydrogen atom towards to oxygen and other hydrogen atoms away from the oxygen. This is anti-symmetric. In other diagram, CO2 For example, carbon dioxide. In carbon dioxide, two types of vibrations also available. So thank you.